And Dana, you can see my slides okay?
I can see your slides just fine. Great.
Okay. Okay.
You show some pictures of your trip. Owen? Another time.
I've only had to sidestep 2 rattlesnakes and 1 Gila monster so far. I know that Liz Gardner, when I saw the rattlesnake, I know Liz Gardner would like me to pin it down and take some of the venom for her.
Okay, I've got half past the hour, so I think we could go ahead and start back up again. Our next presentation is going to be given by Owen McCarty, who very kindly has stopped in the midst of his, I think, 475-mile trek across Arizona to get himself into a Holiday Inn Express with some Wi-Fi and be able to participate in this symposium today. So thanks very much for breaking up your holiday for us, Owen. We really do appreciate it. For those of you who don't know Owen is a professor in the Department of Biomedical Engineering and also the Division of Hematology and Oncology at the Oregon Health Sciences University School of Medicine. He is the chair of the bioengineering department, and he's also a fellow of the American Heart Association. And Owen's going to give us quite an interesting talk about something near and dear to Ed's heart. So Owen, go right ahead.
Great. Thank you very much for the kind introduction. So the Haida are one of the indigenous peoples of the Pacific Northwest. That's where Vancouver now stands. And within the Haida narrative, the raven is a central character. The raven embodies a magician, a transformer, a potent creative force, a trickster, but always a hero. For it was the raven that actually released the first humans from underneath the cockle shell on a beach. And that is a story depicted here by Bill Reed, a luminary artist who dedicated his work to celebrating the diverse indigenous cultures of the Northwest Coast. I was honored to receive a copy of this very work as a gift from Ed for speaking at the Earl Davies Symposium back in 2015. This raven is one of the focal points of my office. It's catalyzed and overheard many discussions: science, careers, life, love, loss. The raven never actually creates anything. They made the world by stealing, exchanging, redistributing, generally moving things around. Perhaps much like my role as a group leader and chair of a department. In learning about the artist, Bill Reed, I was struck by one of his quotes. "Joy is a well-made object, equaled only by the joy of making it." And that is why we gather here today, to celebrate Ed Conway and his leadership, to reflect on the discoveries that he's made, as well as the joy he had and brought to us while making them. Ed's scientific career started under the mentorship of Dr. Bob Rosenberg. Efforts in his lab led to the cloning of thrombomodulin as part of his quest to develop sensitive and specific assays to define the pre-thrombotic and hypercoagulable state of humans. Early work by Ed, working with Dr. Rosenberg, was the first to show that the predominant suppressive effect of the monokine TNF-alpha on thrombomodulin expression by endothelial cells is at the level of transcription. And actually, this suppression occurs independently of de novo protein synthesis. A memoriam honoring Dr. Rosenberg and his career was written by Hugo McCarty, Ken Bauer, and Monty Krieger, and appeared in JTH in 2020. The work done in Ed's lab has gone on to shed light on some of the mechanisms by which thrombomodulin catalyzes anti-inflammatory as well as anticoagulant activities by serving as one of the sites of catalysis for activation of protein C as well as TAFI. This work then led to the major clinical discovery in which his group sequenced the entire thrombomodulin gene in patients with atypical hemolytic uremic syndrome as compared to healthy controls. They found that thrombomodulin variants associated with this disease had diminished capacity to inactivate C3b of the complement cascade, and we'll talk much about this, and to activate procarboxypeptidase B, and thus were protected from activated complement. As an essential arm of the innate immune system, activation of this complement system proceeds via 3 pathways as depicted here. On the left, the lectin pathway, the classical pathway, and alternative pathway, which then converge to form C3 convertases and cleave C3 into C3b and C3a. This system is classically understood to be triggered by exposure to danger signals or damage-associated molecular patterns of, for example, pathogens, DNA, and damaged host cells. Work from Ed's group has made pioneering discoveries into how the complement system, perhaps very much embodying the raven, plays a role as a magician, the transformer, the potent creative force, and sometimes trickster when it comes to interacting with components of the circulation, including the endothelium, regulation of von Willebrand factor polymerization, activation of platelets and leukocytes, and interplay of both the extrinsic and intrinsic pathways of the coagulation cascade. Always a hero. The classical pathway of the complement system is activated as a byproduct of the contact activation pathway, in which the serum protease, factor 12A, activates C1R and C1S to their active forms. The contact pathway of coagulation is initiated in part when blood encounters foreign artificial or biological surfaces, therefore leading to platelet activation, fibrin generation, perhaps to sequester and perhaps even isolate challenges as an extension of the innate immune system. That was why we were surprised by an observation we made during a study when we were examining the effects of inhibiting the ability of factor 12 to activate factor 11 by an antibody, 3G3, depicted on the right-hand side there, on preventing organ damage and death of baboons that were challenged with Staph aureus. And you can see on this graph here, we're showing that the treatment with the red line there, with the Staph aureus would cause organ damage and death, and this was completely inhibited by inhibition with the 3G3, as that dotted line shows. During that study, perhaps fortuitous, we found that blocking factor 11 activation downstream of factor 12 resulted in the blunting the actual blunting of the complement cascade activation, in particular C3b and C5b9. This suggests there exists some crosstalk between the complement pathway and the contact activation system of coagulation beyond the ability of factor 12A to activate C1R and C1S to their active forms as described earlier. This is the light in the fact that 3G3 blocks reciprocal activation. This is depicted on the bottom of the slide here. 3G3 blocks the reciprocal activation of the coagulation factor 11 downstream of 12A. We were curious whether this hinted at a direct or perhaps indirect mechanism of action for factor 11. We therefore looked at all the known substrates of factor 11 to determine whether any of the members of the complement cascade exhibited sequence homology for a known factor 11 cleavage site. And indeed, complement factor H is the major inhibitor of the alternative pathway of the complement system and is structurally related to beta-2 glycoprotein 1, which itself is known to bind to and act as a substrate for factor 11. We therefore set forth to determine whether factor 11 interacts with and potentially neutralizes factor H. And this work was conceptualized and driven by my colleague and junior group leader at OHSU, Dr. Christina Puy. Now, as background, Factor H is composed of 20 homologous complement control proteins, acronym CCP, domains known as short consensus repeats or sushi domains. The 4 N-terminal CCP domains bind to C3b and are necessary for cofactor activity, whereas the CCP 6 to 7 domains and the CCP 18 to 20 domains are responsible for binding to glycans and facilitate the regulation of complement activity on cell surfaces, such as endothelial cells as shown here. And now in solution, factor H enhances the dissociation of convertase complements from C3 convertase as part of the process termed and shown here as the decay accelerating activity. Moreover, factor H provides a cofactor function by binding to C3b, which provides a platform for factor I to bind to factor H C3b complex and cleave C3b into fragments that can no longer form the C5 convertase in C3 convertase. Now, factor H actually circulates in the plasma at a concentration of about 500 micrograms per ml and has a molecular weight of about 150 kilodaltons. The liver, that's a major source of where factor H is formed, yet it can be secreted by endothelial cells, platelets, monocytes, as well as fibroblasts. The critical role of factor H is in normal physiology is actually exemplified by the fact that mutations in the factor H gene and autoantibodies against factor H are associated with atypical hemolytic uremic syndrome, age-related macrodegeneration, dense deposit disease, and antiphospholipid syndrome. So in terms of the science, we then first set out to determine whether factor 11A as an enzyme cleave factor H over time. And as shown here on the left, bottom left here, we observed a degradation of factor H over time to 2 smaller fragments, a 130 Dalton kilodalton band, labeled here, labeled fragment 1, you can see that band showing up, and a smaller, about 35 kilodalton, lower on the gel, 35 kilodalton band, here labeled fragment 2. And then when we blotted using antibodies that specifically recognize either the N-terminal or the C-terminal domain of factor H, we found there that a factor, indeed, the fragment 2 was only recognized by the N-terminal antibody, while the C-terminal antibody only recognized fragment 1, confirming that indeed factor H was being split and cleaved by factor 11A in this purified system. So we next went in there and actually cut out the fragment 1 band to take it for N-terminal sequencing. And amino-terminal sequencing of the bands under reducing conditions revealed that the cleavage of factor H by factor 11A occurred at the end of the CCP 6 domain at the arginine 341-342 domain. We did check to see if other coagulation proteases also cleave factor H. Neither factor 12A, factor 10A, or thrombin cleave factor H. Kallikrein, it was somewhat able to cleave factor H, but at a much lower rate than factor 11A. Now we did some functional studies. We next examined whether factor 11A then neutralized factor H and had functional effect on the role of factor H as a cofactor for factor I degradation by C3b, a potentiation of C3b binding to cells, and dissociation of C3 convertases as our 3 functional assays. And as shown here, I'm just showing some data from these experiments. Indeed, the degradation of factor H by factor 11A decreased the capacity of factor H to promote the cleavage of the alpha chain of C3b. It decreased the binding of factor H to endothelial cells and reduced the capacity of factor H to enhance the decay of C3b. And you can see that in the various plots here, where the vehicle is in the black bars and the open circles are with factor 11A. You can see that decrease both in the binding to endothelial cells and the decay acceleration. Now, to determine whether factor 11A was also able to cleave factor H in a more complex system, We then next studied whether factor 11A can cleave factor H that is bound to the surface of endothelial cells, denoted here as HUVECs. As shown in panel A on the left, we observed that after incubating HUVECs with factor 11A for about 30 minutes, the level of factor H that was actually bound to the cell surface of endothelial cells diminished in a factor 11A concentration dependent manner. And that can be seen as you disappear that band at the 170 kilodalton level. Moreover, we observed that factor 11A was able to cleave factor H present in the releasate of activated platelets, also shown here is in panel B. And we next determined whether factor H can be cleaved in plasma system by factor 11A solely generated by activation of the contact pathway of coagulation. So in all the previous work, we're actually adding in factor 11A. And here we're going to generate it in the plasma to see if endogenous generation of 11A can still have these effects in plasma. And that's what on panel C we're showing here. So human plasma was incubated with an APTT reagent. That's going to activate factor 12 for about 1 hour to induce the generation of factor 11A as a result of activation of factor 12. Now, as shown as panel C, the addition of this APTT reagent to the human plasma induced the generation of that 130 kilodalton fragment H fragment. That's indicative of that degradation of factor H. Now, in the sample where the APTT reagent was incubated in factor 11 depleted plasma, that appearance of the 130 kilodalton band was dramatically reduced, and that's that third column there. And then if you reconstitute the factor 11 depleted plasma with recombinant factor 11, once again, you induce the appearance of that fragment of factor H. So just by activating factor 12 to 11, and now 11A being generated, now you can degrade factor H in plasma. So in summary, we found that factor 11A cleaves the complement factor H at the arginine 341 and 342 bonds at the end of the CCP6 domain of factor H. We found that factor 11A as an enzyme actually reduces factor H cofactor and decay accelerating activity and the binding to endothelial cells in terms of function. In work I didn't have time to present here, we also interestingly found that factor H binds factor 11 and inhibits 11 activation by either thrombin or factor 12A. So there is a loop that's being formed here. And our study now provides a novel molecular link between the contact pathway of coagulation and the complement system. And these results suggest that 11 generation enhances the activity of the complement system and thus very well may potentiate the local immune response. In this work, led here by Christina Pitchard and Christina Pui, has been recently accepted to the Journal of Immunology. I must congratulate Christina for both of getting this paper accepted as well as starting something new here during the shutdown. Quite a tremendous feat. I want to thank all my colleagues that are listed here as co-authors for this rich and exciting collaboration. And certainly a lot of the impetus came from that very conversations with Ed and Earl back in 2015 at the symposium. So perhaps after all, factor 11 is our raven, a trickster, mischievous, an instigator, implicated in disease, yet we fear her absence. Perhaps factor 11A plays its most sinister role by lifting the shell of inhibition off of the coagulation and complement pathways and inactivating negative regulators, including TFPI, Adams TS13, and herein now complement factor H. And these findings should be taken into account as we design drugs to clip the wings of our hero in an effort to safely treat the pre-thrombotic or hypercoagulable state of humans. Thank you again, Ed, for the honor of inviting me to speak at the symposium. It's been a career and personal highlight for me to get to know you and work alongside you. And I'm free to take any questions at this time.
Thank you very much, Owen. So for questions, you can either put them in the chat box or feel free to put your hand up. Ed, would you like the first
Love
hit?
to. Owen, that was fantastic. I love it. I do have a couple of questions. For one, just to point out, arginine 341 has been associated with atypical HUS. Although I don't know that the functional, the mechanism by which it causes atypical HUS, which is not infrequently the case, has been identified. So this would really be interesting to potentially ascribe it to a resistance to cleavage by factor 11A. So I think it's really cool. Now, we showed that factor H interacts with polyphosphate way back. We really never got around to figuring out the relevance. And do you have any idea? Because certainly poly P interacts with 11A, as I recall. It's been a little while since I looked at that, but yes. Well, I'll leave it with that question in particular.
Yeah, that's really good comment. So we already looked at the ions in terms of zinc and manganese and other ions to see if that potentiated. We didn't see any effects there, but we're currently looking at both short and long chain polyphosphate to see if this would further catalyze. Seeing that catalyzes the activity of 11A, would this then have that effect, especially as it's being secreted out of platelets or on maybe perhaps on a platelet surface. Yeah, I see a question in the chat from Laura Healy. Yeah, she mentioned the divalent cations, but yeah, that's what we looked at. We had a hunch that we'd see some maybe zinc play a role here, but in our assays, in our hands, under conditions, we didn't see any of that effects so far.
Well, I'll jump in with another question if there's no other questions there.
Please
so
do.
factor H in COVID. Have you guys looked at all? Because we've identified some patients with changes in factor H levels. We haven't done functional assays, quite frankly, to look specifically at factor H. But have you done any work in that way? And in terms of that, also in terms of factor 11A levels?
Yeah, you know, so we've been working with Alvin Schmeier on this, trying to get some patient samples, and everybody here understands the difficulty when patients may have been already treated with something. We've yet to kind of find that really smoking gun of 12 activation, robustly activated in the patient samples that we were analyzing. So we started looking, and we've been looking for a real rationale for then putting in antibodies against either 12 activation or downstream with the 11 activation. But the signals have been pretty low, and we were a little bit surprised because we thought, especially when the initial the numbers were coming out with some of that clotting, we thought that this would be certainly that 12 and 11 would play a role there, and we've yet to really see that materialize.
Do we have any other questions?
I have to just you know, and I have to also thank my colleague Floria Lupu, who's an expert in complement, and actually in our baboon models where we challenged with staph, and we were looking at, we've had 2 studies now, one blocking the activation 11, we have a second one where we blocked the activation of 12, activity of 12, and we actually ran the complement, seeing Floria was a expert in complement, we ran that as our control, thinking that that's going to stay standard. And we saw that complement really drop off. We were stunned and thought, especially when we were blocking 11, how is this now feeding upstream? And it really challenged our paradigm. And then that's when we had to really dig into the work that Ed and his team have led in that area, that crosstalk between 11 and 12.
Great. Thank you very much, Owen. I think we might as well take advantage of the fact we have a couple of extra minutes and move on to the next speaker, who's Dr. Patricia Liaw from the Department of Medicine Hematology Division at McMaster. She holds the Jack Hirsch Professorship in Thrombosis, and she's also the chair of the Canadian Critical Care Translational Biology Group. Her research focuses on understanding the biological links between innate immunity, blood clotting, and immunothrombosis. And her approach to much of this is to integrate lab-based studies with animal models, as well as with translational research to better understand the pathophysiology of thrombosis, sepsis, and DIC. So Patricia, thank you very much for agreeing to join this seminar today. It's over to you.
Thank you very much, and wanted to thank Dana as well as Ed for the invitation to speak here. It's a honor and privilege to be able to share the work that we've been doing in the field of sepsis and more recently in patients with COVID-19 infections. Can everyone hear me okay? Okay.
Yes, we can.
So I first met Ed years ago at one of the Margaux conferences on critical illness. And unfortunately, it wasn't held in Margaux, France. I'm not sure why, but they actually moved it to Atlanta. But it was a great opportunity to learn a little bit more about the interface between inflammation, coagulation, and the endothelium. And at the time, I was a new postdoctoral fellow with Chuck Esmond. And so it was nice to meet some of the key scientists in the field, especially Ed, who was a fellow Canadian. And one of the things that impressed me most about Ed was that he was extremely kind, and he also had the ability to explain very complicated concepts in clear and simple ways, which I really appreciated as a new trainee. And even though the term immunothrombosis wasn't yet introduced at the time, Ed was one of the key researchers to identify the critical links between innate immunity, clotting, and the endothelium, particularly related to thrombomodulin and the protein C pathway, which we just heard from Owen. And just going through the participant list, I noticed that Nigel Mackman and John Griffin are here as well. So just want to say hello to them as well. So over the years, we've been fortunate enough to meet Ed at various meetings. I think we've hosted him at TARI and McMaster a few times as well. The last in-person meeting, I believe, was ISTH in Melbourne. And also wanted to give a special thanks to the Center for Blood Research for generously sponsoring 7 trainees from McMaster to attend the Earl Davies Symposium back in 2019. So that was a very generous opportunity, and just wanted to say thank you for that. Also, Peter Grin, who's shown in this group photo, he did his master's at McMaster, and he's currently a PhD candidate at UBC. So wanted to say hi to him as well. So our research program focuses on the mechanisms of thrombosis in small vessels related to sepsis and disseminated intravascular coagulation. We also study the role of the vascular endothelium in regulating clotting and fibrinolysis. And we identify the role of immune cells in the process of immunothrombosis. So this was a term that was conceptualized by Engelman and Masberg in 2013. It describes the active participation of the innate immune system in forming thrombi in response to pathogens, as well as self-danger molecules, also known as danger-associated molecular patterns, or DAMPs. This is a really nice diagram which shows the key ways in which the innate immune response can trigger immunothrombosis. So the pathogen-associated molecular patterns, as well as the DAMPs, can induce tissue factor expression on monocytes and also release tissue factor-bearing microparticles. The components of neutrophil extracellular traps, such as neutrophil elastase, can inactivate natural anticoagulants, including TFPI, as well as antithrombin. The Willebrand factor that is bound to NETs, as well as the histones, can recruit and activate platelets. Platelets themselves can facilitate the process of netosis, and also poly-P released by activated platelets can activate clotting. And finally, complement, particularly C3A and C5A, can activate platelets as well. So in our lab, we're interested in NETs as a double-edged sword. It does provide protective effects, specifically through the release of chromatin into the extracellular environment. And that serves to trap pathogens, but it also provides a very high local concentration of antimicrobial peptides that aids in pathogen clearance. However, the major structural component of NETs, which is DNA, can induce harmful effects because it has the ability to activate factor 12 and 11 of the intrinsic pathway of coagulation. And we've also shown that DNA can inhibit fibrinolysis by inhibiting plasma-mediated digestion of the fibrin clot. Histones, which are the major proteins or the most abundant proteins found in NETs, can impair anticoagulant pathways. Specifically, it binds to thrombomodulin as well as protein C, thereby inhibiting APC generation. They are cytotoxic to vascular endothelial cells, and they can induce procoagulant activity through activation of platelets and also on red blood cells. So given the importance of NETs in enhancing clot formation, it's logical to consider dismantling NETs with DNase 1. So recently we looked at endogenous DNase 1 levels in septic patients, and this is part of Sahar Sarabipour's master's thesis. And she found that compared to healthy controls, the levels of DNase 1 are persistently low throughout the course of the patient's stay in the ICU. She also found that heparin has the ability to enhance the digestion of NETs by DNase 1. And by using different heparin variants, she was able to show that the enhancing effect is dependent on the size of the heparins and independent of its anticoagulant activity. So these are immunofluorescent images of NETs released by activated neutrophils. And you can see that when you add DNase 1, there is partial digestion of the NETs, which is then enhanced further by either unfractionated heparin, enoxaparin, or Vasoflux, which is a non-anticoagulant heparin. In contrast, fondaparinux, which is the smallest heparin, doesn't have the ability to enhance DNase 1 mediated degradation of the NET scaffold. So this is work that we recently submitted and is currently under revisions. This is a graphical abstract that Sahar has produced to show that there are different ways that you can loosen DNA histone complexes, either through displacement of histones by heparin variants or through PAD4 mediated modification of histones. And both ways can result in a looser DNA histone complex, which then is more susceptible to digestion by DNase 1. We also studied novel therapies for sepsis using a mouse model of cecal ligation and puncture. And we've looked at DNase 1, also known as palmazine, and different heparin variants. The analysis is through survival of the mouse upon induction of sepsis, and organ histology is examined. We also look at markers of inflammation, blood clotting, as well as fibrinolysis. One of the first papers that we published in this field showed that IP administration of DNase 1 not only reduces organ damage, but it can improve outcome in a severe model of sepsis. So these were 24-hour studies where we had 100% mortality after 24 hours. These graphs show time-dependent changes in extracellular DNA, as well as IL-6, IL-10, and thrombin-antithrombin complexes in our mouse model of CLP. More recently, we've extended the studies to a more moderate model where we're looking at survival up to 72 hours. And these are studies that are part of Sarah Madero's PhD thesis. And she's made an interesting observation that DNase 1 appears to improve survival in septic mice. Similarly, we've also looked at Vasoflux, which is a non-anticoagulant heparin. And at a dose of 1 mg per kg per 24 hours, we found that there also is a survival benefit. And these are studies that are done by Neha Sharma. So in parallel with the animal studies, we've also been conducting multi-center translational studies, and with support from CIHR, we've actually conducted one of the largest pan-Canadian translational studies of ICU patients. We've collected clinical data as well as blood samples from almost 400 hospitalized patients with sepsis, as well as 362 non-septic patients. And this has allowed us to explore biological mechanisms related to the prothrombotic state in sepsis. So as an example, we've looked at hemostatic markers that can be useful for defining the pre-DIC state. This is a multi-center observational study which showed that platelets in INR in combination with protein C as well as antithrombin can identify the pre-DIC state. In contrast, we found that increases in D-dimer and decreases in fibrinogen are observed in the later or overt stages of DIC. And the data also suggests that NETosis contributes to the onset of DIC in sepsis. So despite numerous phase 3 clinical trials in sepsis, currently there are no sepsis-specific therapies. And what this review article showed was that there have been over 60 clinical trials performed involving over 22,000 patients. These interventions included anti-inflammatory agents as well as anticoagulants. And even though the preclinical animal studies as well as the phase 1 and 2 studies look promising, the results were negative in phase 3 randomized controlled studies. So the question is, are there ways to improve clinical trials in sepsis where we can include prognostic enrichment as well as predictive enrichment as part of the trial design, the way that it is done in the field of oncology, for example? So the enrichment strategies are supposed to be helpful for selecting patients in whom an intervention is more likely to be effective. However, there are major knowledge gaps, not only for prognostic enrichment, which allows a stratification of patients depending on their risk, but also knowledge gaps for predictive enrichment based on differences in biological mechanisms. So for the past several years, we've been looking at potential mediators of death in septic patients. And we've identified 6 main indicators that appear to collectively be useful for predicting poor outcome in patients with sepsis. And these include elevations in extracellular DNA, which we mentioned earlier can not only activate clotting, but at sites of microvascular thrombosis, they provide a scaffold for red blood cells as well as platelets. There's also decreased levels of protein C due to consumption and decreased levels in platelets, which reflects disseminated intravascular coagulation. We've also looked at elevations in lactate, which is a marker of tissue hypoperfusion, and increases in levels of creatinine, as well as abnormalities in the Glasgow Coma score. So this work was registered with clinicaltrials.gov. It's called the Dynamic Study. And the overall objective was to develop a personalized tool for assessing mortality risk in septic patients. And our hypothesis was that the combination of these 6 indicators can not only predict mortality, but may be useful to generate personalized mortality risk profiles. So this was a prospective multi-center observational study of almost 400 patients from 9 ICUs across Canada. We collected an extensive amount of clinical data and longitudinal blood samples for the duration of the patient's stay in the ICU. So this was published on behalf of the Canadian Critical Care Translational Biology Group and the Canadian Critical Care Trials Group. This shows an example of how the algorithm can be used for a particular patient. So we would input the levels of the indicators on day 1, so on admission to the ICU, and we also would input how these indicators change over time. And for this particular patient, you can see that having high levels of DNA on admission and very low protein C and platelet counts would increase the risk of dying within 28 days. So we found that the predicted probability for this particular patient is 97%. And you can also predict the probability of dying on any particular day. So for example, if we picked day 11, then the probability of dying would be 12%. This pie diagram shows the relative predictive powers of the 6 prognostic indicators. And what I wanted to highlight is that the ability of these indicators to predict poor outcome varies significantly. So for some indicators, most of the predictive power comes from the day 1 values. For example, levels of extracellular DNA. And there's very little predictive power based on the change in the levels over time. And that's because for this particular marker, the levels are persistently elevated throughout the patient's stay in the ICU. In contrast, a marker like protein C or platelets, there is predictive utility for using the day 1 values, as well as how the markers change over time. This is an example of a personalized mortality risk profile that highlights the relative contribution of each indicator to the risk of dying. So for this particular patient, it suggests that the abnormalities in protein C and platelets appear to be the major contributors to the patient's risk of dying. So the relevance would be that this patient may benefit from anticoagulant therapies. So our vision is to have a precision medicine approach for septic patients, where we're able to have a point of care test where the levels of the 6 indicators are analyzed through a risk assessment tool, and the output would be the probability of dying on a specific day, and also the identification of which indicators contributes the most to mortality risk. The applications then would be to partner this precision medicine approach with a targeted therapy, and also be able to monitor response to interventions. So then the enrichment strategies would be applicable at the level of prognosis and also in terms of predictive enrichment, which would be individualized for each individual patient. So in the next part of my talk, I just wanted to switch over to some of the recent work that we've been doing, looking at immunothrombosis biomarkers for distinguishing COVID-19 patients from non-COVID septic pneumonia patients, and also for predicting ICU. And this was done in collaboration with the COVID Beacon study, which is co-led by Paul Kim as well as Calvin Ye, and many investigators at TARI, as well as within the Canadian Critical Care Translational Biology group and the trials group. So in terms of thrombosis in COVID-19, this is a slide that is courtesy of the ATTACK website, where they reported the results of their interim analysis. So thrombosis is a prominent feature, with 5 to 30% of patients developing thrombosis based on studies by Tang and Clark et al. Venous and arterial events have been reported, and microthrombosis may be a key mediator of COVID-19 related organ dysfunction, morbidity, and mortality. This highlights the proposed mechanisms of COVID-19 associated thrombosis. And I just wanted to highlight that this is very similar to what I presented a little bit earlier in the talk in terms of the links between infection, clotting, endothelial dysfunction, and microvascular thrombosis. So the objective of our study is to identify biomarkers that can distinguish between COVID-19 patients and non-COVID hospitalized patients with pneumonia. And the reason for comparing pneumonia patients is because sepsis is an extremely heterogeneous condition. So you could have abdominal sepsis or urosepsis, but we thought that by focusing on ventilated patients with pneumonia, we may be able to identify pathways that are unique to COVID-19 infection. The secondary objective is to evaluate the utility of the biomarkers for predicting ICU mortality in the 2 patient groups. So this is a hypothesis generating study. The sample size is very small. We have 14 COVID-19 patients with 50% mortality rate, as well as 19 age-matched non-COVID septic pneumonia patients, and the mortality rate there is 32%. The plasma samples were collected longitudinally, so up to day 10 for the COVID patients, and up to day 14 and beyond for the non-COVID septic pneumonia patients. And we also had healthy age and sex match controls. The COVID patients were kindly provided, the plasma samples from the COVID patients were kindly provided by Doug Fraser and his colleagues at London. And the non-COVID patients are part of our dynamic studies. So we have a very large existing biobank of these samples. We looked at immunothrombosis biomarkers, and we divided them into markers of coagulation. So that would be thrombin and antithrombin complexes, natural anticoagulants, which include protein C and antithrombin, fibrinolysis as measured by D-dimer and fibrinogen, netosis, which we measured through levels of citrullonated histone H3, as well as extracellular DNA, and then soluble EPCR and soluble thrombomodulin as indicators of endothelial dysfunction. The baseline characteristics of the patients were very similar in terms of the MOD scores, SOFA scores, and some of their comorbidities. 100% of the non-COVID septic patients were ventilated. And for the COVID patient, I think around 80% were ventilated as well. So this compares the coagulation, anticoagulant profiles longitudinally between the 2 patient groups. What I've shown here in the gray are the levels in healthy individuals. So if we look at fibrinogen, elevated levels of fibrinogen is a hallmark of COVID, which is something that many other groups have reported as well, and significantly higher than in septic patients with pneumonia. What we found interesting as well is that in terms of antithrombin, the COVID-19 patients had levels of antithrombin that were within the normal range, whereas protein C levels for both groups were around 50%, which is what you would expect in terms of consumption of anticoagulants, particularly the protein C pathway, which plays a major role in inhibiting thrombosis in the microcirculation. So this observation, I think, is relevant to the work done by Doug Fraser's group, where they showed that endothelial injury as well as glycocalyx degradation is a prominent feature of patients with coronavirus disease 2019. In that paper, they found that patients with poor outcome had increased levels of syndicate-1 as well as chondroitin sulfate. So these are part of the glycocalyx, which provides a thromboresistant surface under normal conditions. And perhaps the near normal levels of antithrombin that we're seeing in COVID patients is because of the lack of the glycocalyx. So if you have shedding of heparin sulfate, then you're missing the catalyst that is required for accelerating the inhibition of thrombin and factor Xa by antithrombin. We found that D-dimer levels as well as soluble TM levels were elevated in both patient groups compared to healthy controls. And for soluble EPCR, the levels in sepsis are actually higher than in COVID-19. Markers of H3-CYT are also elevated in the 2 patient groups compared to healthy controls, as well as circulating DNA. What was interesting is that once we separated the 2 patient groups further into survivors and non-survivors, we found that, well, first of all, we found that there are 4 biomarkers that can distinguish COVID-19 patients from non-COVID septic pneumonia patients based on ROC analysis. And these include fibrinogen, soluble EPCR, antithrombin, as well as circulating DNA. And then we separated the 2 patient groups further into survivors and non-survivors. what was interesting is that the predictors of ICU mortality is actually quite different between the two patient groups. So for COVID-19, the predictors of ICU mortality are soluble thrombomodulin and citrullonated histone H3. For pneumonia patients, it's protein C as well as increases in extracellular DNA. So in summary, we found that 4 markers have utility for distinguishing COVID-19 patients from non-COVID septic pneumonia patients. And the main predictors of ICU mortality are different. So soluble TM and H3 CYP for COVID patients, DNA and protein C for septic pneumonia patients. And so this hypothesis generating study suggests that the pathophysiology of immunothrombosis differs between the two patient groups. And we do plan to validate the findings prospectively in a larger cohort of patients. The final part of this presentation, I just wanted to highlight work that we've been doing on behalf of Sepsis Canada and also the Canadian Critical Care Translational Biology Group. This is the National Preclinical Sepsis Platform with co-leads being Manoj Lalu, Asher Mendelson, and Braden McDonald, and many members of the Canadian Critical Care Translational Biology Group as well. So one of the knowledge gaps that has been identified in terms of preclinical research is that there are several methodological challenges. So if you think about clinical research, they're often multi-center, they're randomized and blinded, they're adequately powered, protocols are shared between centers, they're standardized reporting, and so it's easier to synthesize the information. In contrast, preclinical research involving animal models are usually single center studies. They're often not randomized, not blinded. There are no sample size calculations. And there's a lot of variation in protocol between labs. So sometimes it's difficult to reproduce findings, and the reporting is often incomplete. So for example, if we consider biological sex, a recent survey done by Manoj Lalu's group found that I think only a handful of studies in preclinical research have specifically looked at the effect of biological sex on outcome. And so the goal of the National Preclinical Sepsis Platform is to harmonize protocols across different centers. So currently we have 10 participating centers within Canada. And the model that we're going to use for starters is the fecal-induced peritonitis model, also known as FIP. So unlike the cecal ligation and puncture model, which we've been using for many years, the FIP model is easier to standardize. And there's also a familiar locus of infection, which would be abdominal. We can monitor disease progression and adapt the amount of, we can adapt the severity of the model depending on how much fecal slurry is actually administered into the animals. We will also include clinically relevant treatments, including the use of antibiotics as well as fluids in this Miri model of sepsis. So the fecal slurry will be frozen and standardized. It'll be made at 2 centers. So at Tari, Dhruva in our lab is responsible for making the inoculum, and we'll be shipping it nationally. And then Stefan Bork over in Edmonton has also produced his own inoculum as well. And there will be a harmonized mouse database as well as a biobank. And this is led by Sean Gill, Braden McDonald, and Janet Sunohara Nielsen. So the standardized protocols would include collection of blood and plasma samples, various tissues, and lavage samples as well. So the scientific goals of the MPSP are to create a multi-center infrastructure to evaluate the pathophysiology of the host response and the biological heterogeneity of sepsis in a controlled preclinical environment. The second goal is to identify research goals that are clinically relevant and patient-centered. And finally, to have adequately powered, high-quality clinical data that will accelerate the movement and translation of novel therapeutics for sepsis into early clinical testing. And just want to end by saying thank you very much to numerous colleagues and collaborators at TARI and McMaster University, as well as within the translational critical care groups within Canada and the trials group, and funding support from CIHR as well as CamVector. And I'm happy to take any questions that you would have. Thank you so much.
Great. Thank you very much, Patricia. This is open for questions. People can either put them up. I see you've got one already in the chat box.
Sure, I see one from, I see one from Colin Krebs,
Yep.
who's asking, do COVID-19 patients in the ICU exhibit clinical features that qualify as sepsis, or is this something different? So yes, to clarify, hospitalized COVID-19 patients, by definition, are septic patients, because sepsis is defined as systemic inflammation and coagulation with at least one dysfunctional organ system. So when we talk about COVID-19 ventilated patients, by definition, they are sepsis already. So when we have the non-COVID group, we're really trying to identify pathways that are unique to the SARS-CoV-2 virus. So to answer Colin's question, do they exhibit clinical features that qualify as sepsis? Yes. A question from Dave Farrell. The high fibrinogen levels suggest an independent mechanism for thrombosis. Paul Kim showed that fibrinolysis shows first order kinetics with fibrinogen concentrations such that the higher the fibrinogen, the slower the fibrinolysis. Do you think that could be playing a role in COVID-19? I think so. So Paul has been using the same sample set that we have been using for COVID-19. And at this point, we haven't looked at the first order kinetics, but I think consistent with what Paul has shown, I think his model makes sense in this context as well.
And I think Peter Grin has a question for you.
Yeah, hi Patricia.
Peter must be unmuted. Hi, Peter.
Hi, great talk as always. It's exciting to see what's been going on at McMaster since I've moved over here to the Center for Blood Research. I just want to ask, are you planning to, once you have data from your COVID prospective study, are you planning to apply the same sort of algorithm from dynamics over to possibly predict how sick patients get from COVID-19?
I think that might be a little bit premature because the algorithm for the dynamic study was based on 400 non-COVID septic patients with different sites and types of infection. So I think we would first have to recreate the algorithm with COVID patients. And if it turns out that some of the indicators are very similar, then I think it would have utility for both. But that's a great question. And I think it's something that we definitely would be interested in exploring, especially now that at the Hamilton General, we've been recruiting patients under the leadership of Paul Kim. So we're hoping that we will get the sufficient numbers fairly soon.
And I suppose one of the things is that with the numbers spiking, it might not take all that long compared to how long it took for the dynamic study with all the septic patients. So.
Yep, that's correct. We also have collaborators with Claudia DeSantos's group in Toronto, and I believe Doug Fraser's group in London is also recruiting as part of the translational group.
All right, thanks, Patricia.
Great. I see Dr. Conway is waving his hand.
Hi, Patricia. That was fabulous. Okay, so I just make a couple of comments, stories, and then a question. One, I used to go to when Chuck Hesman was around in his earlier days. I used to go to his lectures, seminars, whatever, and I was always blown away. Hey, he doesn't have a medical degree. How does he know all this stuff? I mean, he sounds like a clinician scientist who's been frontline. And it strikes me that Patricia trained with Chuck, and she has the same amazing talents. She unless she's been training on the side, I don't think she really has a medical degree, but she's really ensconced herself into this field, and she's surrounded by a fabulous center. So I would just kudos to you for all that you've done. The second little story is the Margot conference. I do remember it really, really well. One, I had more hair, but aside, I was quite young, relatively speaking, and I was invited from Belgium to go to that Margot conference, which was a fabulous conference. And in those days, the conferences really paid well to take you to these conferences. And I flew first class from Belgium. I had never been on a first class flight. And the guy beside me in first class was, stretching out. He opened up the chair so he could, lounge and go to sleep. So I thought, oh, I didn't know anything about this. So I thought, oh, this is a good idea. So I'm sort of fumbling with my right hand, trying to get my seat to go back, and nothing was working. And then I turned to my right and I could see this guy. I had totally collapsed him, controlling his chair. So that's why I do remember the Margot conference. Aside from there was great science to follow. Okay.
What's the fact that it wasn't actually held in Margot, right?
No, it was in Atlanta, right?
Yes.
Great. That's why I remember it well. Okay, third, science. Well, a couple of questions. One, the heterogeneity, which is always a battle in sepsis, it really underlines the challenges you have in animal modeling, doesn't it? Because you use the cecal ligation, which gives you a readout, and it gives you some mechanistic insights. But can you tweak these models to, so that they more clearly represent human disease? And then there's just a second easier question probably is glycocalyx. When it's ripped off, does it expose tissue factor? Does it decrypt tissue factor?
I'm not sure if tissue factor is decrypted. My understanding is that if you strip away the glycocalyx, I don't think endothelial cells in vivo even express tissue factor. My understanding is that it's expressed by subendothelial cells, or maybe there is some, but it would be in a cryptic state. So we haven't actually looked at that. Your first question is related to our model of cecal ligation and puncture. You're right, there are many limitations to that model, including the fact that we typically use healthy adult mice, which are, ages, I think 8 to 12 weeks. And the challenge of having older mice is that you need to pay for housing, you need to age them for 2 years. So that's just extremely expensive. So I think, looking at healthy mice versus what happens in the critical care population, where they're typically older patients, that in itself, the age, I think, is going to be an issue. We can vary the severity of sepsis. So as I mentioned, we have an acute model where we ligate and extrude more of these fecal contents, or the contents of the cecum. And then we have a milder model where the mice are able to survive out to 72 hours. At least, I think 30% of them do survive. But apart from that, it is a model of polymicrobial sepsis, abdominal sepsis. So you're right, it wouldn't really mimic the pathophysiology of like ARDS or infections of other areas of the body. So that is one of the limitations. But I think currently it is considered one of the gold standards in mirroring sepsis research.
Great. Thanks very much, Patricia. I just have a couple.
Question is Jose here?
I think so.
I just want to know what that creature is that he's got.
Oh, good. Can you hear me?
Yes,
We can.
Yeah.
Yes, that creature is a troll. It's called the Fremont troll. It sits under a bridge in Seattle and captures cars in the evening.
The ones that fall into the big dig, right?
Exactly.
Yeah, Jose like a monster.
It's good.
Well, we have something in common.
You're hiding under the bridge.
Does
I'm a
anyone
monster.
just feel free to unmute because it's hard for me to read this this chat.
Yeah, so just to mention the chat, we've just put into the chat a little bit of advertisement. Next month, we're hosting our annual Norman Bethune Symposium. So it's, again, another free symposium. People can feel free to sign up for that. Christina has put the link there. And before we go on our next break, I thought what we might do is ask people to please open up your cameras, because Christina would like to take some screenshots so that we can use them on our website. So would be great to actually see smiling faces instead of just lists of names.